Welcome back to the Deep Dive. Today, we're tackling a question that, on the surface, feels almost philosophical, but it's actually deeply biological. How does your brain maintain a consistent narrative of reality? We all just take it for granted. You walk into a room, you know where you are, you remember why you're there.
And you can filter out the noise, the hum of the lights, the refrigerator. It all just feels seamless.
But for millions of people living with schizophrenia, that seamless narrative just fractures. The filter breaks.
And for decades, the story we've told ourselves about why it fractures has been, well, it's been dominated by one chemical, dopamine.
Right. It's always the dopamine story. Yeah. But today we are pushing dopamine to the side. We're looking at a brand new paper. It was just published in Neuron, February 2026, by Schlegel and colleagues.
And this paper, it reads less like a medical report and more like a forensic analysis, a molecular crime scene.
I like that. So what's our mission for this deep dive?
We're basically going on a detective story. The researchers have found a specific molecular saboteur. And it's not a neurotransmitter. It's a protein that physically hijacks the brain's machinery.
The paper's title is a bit of a mouthful, Elevated Calneuron 1. Something.
Yeah, let's just call it the case of the molecular bouncer.
Okay, the molecular bouncer. So our goal today is to trace this crime from like a single molecule all the way up to the actual experience of cognitive problems.
We're going to identify the culprit, understand the weapon, and then, this is the really crucial part, see how a new class of drugs basically acts as a crowbar to fix the whole mess.
And this is so important because we're not just talking about some vague chemical imbalance.
No, not at all. We're talking about a specific mechanical failure point in a very specific neural circuit. You are going to understand exactly how it breaks.
All right, so let's get to the crime scene. Where are we in the brain?
We are in the prefrontal cortex, the PFC.
The CEO of the brain, right?
Yeah.
Executive function, planning.
Exactly. And the specific victim at this crime scene is a receptor, the muscarinic M1 receptor.
Okay, stop there. Why the M1 receptor? I thought we'd be talking about dopamine receptors.
And that's the key context. Current drugs, the antipsychotics, they are great at blocking dopamine, and that helps with the so-called positive symptoms, the hallucinations, for example.
But not the other stuff.
Exactly. They barely touch the cognitive deficits, the brain fog, the memory loss, the inability to plan. That whole side of the equation is run by a different system, the cholinergic system. And the M1 receptor is the workhorse there.
So the M1 receptor's main job is to help the brain learn and adapt.
Precisely. It enables something called synaptic plasticity, specifically a process called long-term depression, or MLTD.
Long-term depression. Sounds bad.
It sounds bad, but in neuroscience, it's essential. It's how the brain weakens certain connections to refine how it stores information. It's like cleaning up your hard drive. If M1 goes down, the CEO can't update the company strategy.
The phone line is down. Okay, so the researchers, they go into the lab, they're looking at post-mortem brain tissue from the dorsolateral prefrontal cortex of patients with schizophrenia. What do they find?
They find the M1 receptors are there, but they're being swarmed. Swarmed by a massive overexpression of a protein called calneuron 1.
Okay, so way too much of this calneuron 1.
Way too much. The levels of the mRNA that makes it and the protein itself were just significantly elevated. And to make sure this wasn't just a fluke in humans, they checked 2 different genetic mouse models of schizophrenia.
The DISC1 and the DF16 mice.
Yep, 2 of the most robust models we have, and they saw the exact same thing.
Wow.
High levels of calneuron 1, specifically in layer 23 of the prefrontal cortex. That is a critical layer for processing incoming information.
So we have our suspect, calneuron 1. Why? What is this thing? Is it even supposed to be there?
Oh, it's a natural protein, but it has a very specific and kind of weird personality. It's a calcium sensor.
Meaning it grabs onto calcium ions.
Right. It has these little molecular hands for grabbing calcium. But, and this is the absolute key, unlike most other calcium sensors that just, float around inside the cell.
Like guests at a party.
Right. Calneuron 1 is what's called a type 2 tail-anchored transmembrane protein.
Tail-anchored? That sounds permanent.
It means it's physically stuck. It's embedded in the membranes inside the cell. It cannot wander off.
So if the cell is a nightclub, calneuron 1 isn't a guest on the dance floor.
No, it's the bouncer.
And it's chained to the door frame.
That's a perfect analogy. And in these patients, it's like the club hired 50 bouncers for one single door. They are crowding the entrance, the M1 receptor, and because they are anchored, they can't leave. It's a permanent bottleneck.
Okay, let's do a quick recap. It feels like we need one. We're about 5 minutes in, so what do we know?
Okay, 1. The cognitive problems in schizophrenia are linked to a failure of this M1 receptor in the prefrontal cortex.
2. Researchers found a massive overabundance of this protein, calneuron-1, in both patients and in mouse models.
And 3. Calneuron-1 isn't a free-floating protein. It's a calcium sensor that is permanently anchored to the membrane right next to the receptor it's causing trouble for.
So we've established the bouncer is crowding the door. The next question is, what exactly is it doing?
Let's get into the molecular mechanism. This is where the paper just gets incredibly detailed, almost like an engineering blueprint.
So to understand the crime, we need to understand the normal routine.
Yeah.
How does the M1 receptor usually work?
It's a G protein-coupled receptor, a GPCR. Think of it as a relay station.
Signal comes in from the outside.
And it passes a message to the inside. When a signal hits the outside of the receptor changes its shape, and that kicks a messenger on the inside of the cell.
And that messenger is a G protein.
A specific one called G-alpha-Q11.
Let's call him the courier.
Perfect. The courier runs off, triggers a release of calcium, and boom, you get neuroplasticity. The brain learns something new.
Simple enough. So where does our bouncer, calneuron-1, interfere?
It binds to the M1 receptor at a very specific spot, the third intracellular loop, or the I3 loop.
That's on the underside of the receptor inside the cell.
Exactly. And the way it binds is, well, it's almost devious. It's a two-step process.
Okay.
Step 1 is what they call calcium-independent pre-association.
Meaning it's latched on even when the neuron is just resting.
Even when you're asleep, when the brain is quiet, the tail of calneuron-1 is already holding on to the M1 receptor. It's just hugging it. It's lurking.
The bouncer's just leaning against the door handle all the time.
All the time. Then comes step 2, the attack. When the neuron actually gets a signal, calcium levels rise just a little bit, and that's the trigger. The functional hand of calneuron-1 clamps down hard.
Why is that the kill shot?
Because the exact spot where calneuron-1 clamps down is the exact same parking spot for the courier, for our G protein.
Ah, it's a game of musical chairs. And calneuron-1 grabs the chair first.
It's competitive inhibition. The fancy term is steric hindrance.
Steric hindrance.
Which just means I am physically in your way. Calneuron-1 is bulky, it clamps down, and there is physically no room for G-alpha-Q11 to dock. So the receptor can be screaming, I have a message! But the courier has been kicked off the loading dock.
This is all happening on a scale of nanometers. How on earth do you see this? You can't just look at it.
No, you need molecular spies. They used a couple of brilliant techniques. The first one is called FREP.
Okay, explain FRET like I'm a very smart high school student.
All right, imagine you have two tuning forks. If you strike one and the other is really close, the second one will start to vibrate a little bit too, right?
Right, energy transfer.
FRT is like that, but with light. You tag the M1 receptor with a protein that glows one color, and you tag the G protein with another. If they get close enough to touch, they transfer energy, and the light actually changes color.
So what did the Farah data show?
It was a smoking gun. As calneuron-1 levels went up, the energy transfer, the color change between the M1 receptor and the G protein just tanked. It went to 0.
The signal was dead.
And then they used another method, PLA, proximity ligation assay.
The glowing dots.
The glowing dots, yeah. This one's even more visual. It creates a glowing red dot, but only if two specific proteins are practically hugging each other. In a healthy brain, you see a few scattered dots.
And in the high calneuron brains.
The tissue was lit up. It looked like a rash of red clusters.
A rash?
And every single one of those red dots was a crime scene. A place where a calneuron-1 protein was suffocating an M1 receptor.
Wow. That's an image that's going to stick with me. Okay, let's recap this section. This is the core mechanism.
Right. So calneuron-1 acts as a competitive inhibitor. It's always hanging out on the receptor. When calcium rises, it clamps down and physically kicks the G protein off.
And the result is that the entire signaling pathway needed for learning gets severed at the source.
Exactly. And that one molecular break is what causes all the chaos in the wider network.
Which brings us perfectly to part 3. From molecule to network. A broken switch is one thing. How does that broken switch lead to the symptoms of schizophrenia?
The paper makes a very direct link. This molecular failure causes a system-wide collapse in the connection between two key brain regions, the hippocampus and the prefrontal cortex.
Memory and control, they have to talk.
And they have to talk in the right direction. The hippocampus needs to drive the conversation. It sends contextual information forward to the PFC.
And in the high calneuron mice.
That forward drive was gone. It was just abolished. They measured something called the spectral density ratio, the SDR.
Think of it as tracking who's leading the dance.
Precisely. In normal mice, the hippocampus leads. In these mice, the ratio collapsed. It was like the PFC just wasn't listening to the memories anymore.
And the rhythm was gone too,
Yeah.
right? This whole theta-gamma coupling part was fascinating. Can you unpack that for us?
Absolutely. Think of brain waves like an orchestra. The theta waves are the conductor's baton. Slow, steady, 4 to 8 times a second. It sets the beat for learning and memory.
Okay.
The gamma waves are the violins. They're playing really fast, processing all the complex details.
And in a healthy brain, the violins watch the conductor.
Exactly. The violins play their fast notes at very specific moments in the conductor's slow wave. We call that phase locking. It's beautiful. But in these mice, the locking was lost.
The conductor was waving the baton.
And the violins were just playing chaotic noise. The orchestra was out of sync.
You keep using the word noise.
Because it's literal. They did an analysis of the neural noise itself, the 1F slope. And what they found was that the brains with high cal-neuron 1 showed a clear tilt toward excitation. The brain was just too loud, too hot.
And why? What's causing the noise?
It's because the M1 receptor, when it's working, helps to drive the inhibitory system, the GABAergic neurons. They're the ones that quiet down all the background static.
But with M1 blocked by the bouncer.
The quieting system fails, the inhibition is gone, so the brain becomes like a room full of people all shouting at once.
And that explains the weird behaviors they saw in the mice. Like the pre-pulse inhibition test.
Right, the PPI test. Normally, if I play a little quiet tone right before a loud, startling bang, you'll jump less.
Because your brain uses the tone as a warning to filter the bang.
Your brain gates the sensory input. These mice couldn't do that. They startled just as much with the warning as without it. The filter was broken.
Which is a classic marker in schizophrenia. Everything comes in at full volume.
Everything. And then there was the cognitive flexibility test, the set shifting.
This one was really telling.
They taught the mice a rule. If you smell cinnamon, dig in that pot for a reward. Simple enough. Then they changed the rule. Now the reward is in the pot where you see a light.
A normal mouse figures it out after a few tries. Okay, cinnamon is out, light is in.
But the cal-neuron mice, they just kept digging at the cinnamon. Over and over and over. They were stuck.
Oh, perseverance error.
Exactly. It's a perfect model for the cognitive rigidity we see in patients. You can't update your internal model of the world because the receptor that's supposed to process the update signal is being blocked.
Okay, let's pause again. Recap of part 3. The molecular blockade, it leads to network chaos.
The hippocampus stops driving the PFC.
The brain's orchestra falls out of sync. Theta and gamma decouple.
And the result is an electrically noisy brain that can't filter information or switch tasks effectively.
It sounds pretty hopeless, like a permanently broken machine.
It really does. But it's not. And that's the amazing twist in the final part of this story. Part 4, the rescue.
And the hero of this story is a drug called xanomeline.
Xanomeline.
Now, this drug is getting a lot of attention right now. It's the key part of KarXT, which just got FDA approval. We know it works in people, but this paper reveals a mechanism that is, honestly, it's pretty surprising.
It is. It's known as an M1 receptor agonist.
Which usually just means it's like a key that turns the receptor on and pushes the button.
But wait, remember, our receptor's button is being blocked by a 300-pound bouncer, so how can just pushing the button possibly work?
You can't ring the doorbell if someone's body is in front of it.
Precisely. So the researchers gave xanomeline to these high cal-neuron mice, and what they found was incredible. The drug acts less like a finger pressing a button and more like a crowbar.
A crowbar.
It physically disrupts the interaction between cal-neuron 1 and the receptor. They went back to that PLA experiment.
The glowing red dots.
And when they added xanomeline, the red dots, the clusters, they disappeared. The drug was breaking them apart.
So the drug forces a shape change in the receptor that physically shakes the bouncer off its perch.
That's the implication. It overcomes the steric hindrance. And the moment cal-neuron 1 was pried off, guess who could come back to the alerting dock?
Our courier, G-alpha-Q11.
The courier could bind again. The signal was restored. They saw plasticity, the MLTD return in brain slices.
And what about the mice? The behavior?
Completely recovered. The mice regained their ability to filter the startle response in the PPI test. They could do the set shifting task again. They could switch from cinnamon to light. And on the network level,
Let me guess. The hippocampus started driving the PFC again.
the directionality came back. The conductor and the orchestra got back in sync.
That is That is really profound. It suggests that this disconnectivity in schizophrenia isn't necessarily permanent damage. The wires aren't cut.
They're just jammed. It's a functional blockade. And if you can unjam it, the capacity for clear thought is still there, waiting to be unlocked.
Okay, let's synthesize all of this. Let's lay out the grand theory this paper presents.
All right. The synthesis is this. In schizophrenia, for genetic or other reasons, levels of calneuron 1 shoot up in the prefrontal cortex.
This protein acts like a molecular squatter.
It camps out on the M1 receptor, creating a physical bottleneck that prevents the G protein from doing its job.
And that single failure kills the learning signal and, crucially, prevents the brain's inhibitory or quieting system from engaging.
The result is a noisy, disconnected cortex that can't sync with memory or filter reality properly.
And a drug like xanomeline works, at least in part, because it acts as a pharmacological crowbar, physically prying that squatter off the receptor and rebooting the entire circuit.
That's the story, from molecule to medicine.
It's an incredible story, but it leaves me with one big nagging question. Why? Why is calneuron 1 elevated in the first place?
That is the million-dollar question, isn't it? Is it just a genetic accident? The gene is a known risk factor. Or, and this is just a thought, is it a compensatory mechanism that has gone horribly wrong?
Compensatory, what do you mean?
Well, imagine the brain senses that it's becoming too excitable, too noisy for some other reason. What if it tries to fix it? It tries to dampen the signaling by producing more of this bouncer protein, calneuron 1.
It's trying to turn the volume down.
Right, it's trying to protect itself. But it overcorrects. It floods the zone with bouncers, and instead of just regulating the signal, it shuts it down completely, a biological tragedy of good intentions.
And that connects back to the finding that it's binding even at rest, when we're asleep.
It implies the blockade is just. It's relentless. It's not just during moments of stress. It is a constant baseline pressure on a person's ability to think clearly.
I want to throw a question out to you, the listener. Think about drug design for a second. Xanomeline is great, but it's a broad agonist. It hits the whole receptor. Now that we know the exact crime scene, that I3 loop, could we be more precise?
What if you could design a molecule that only targets that one binding site? A little molecular wedge that just pries the bouncer off without activating the whole receptor in other ways?
You might get all the cognitive restoration with far fewer side effects.
It's a whole new way of thinking about treatment.
And it makes you wonder, could other cognitive diseases, like Alzheimer's, be similar? Not broken receptors, but just other receptors being blocked by other proteins. Molecular crowding as a disease mechanism.
I think we are just scratching the surface of protein interactions as a field for new medicines.
Well, we've gone from a mouse digging for a treat all the way down to the steric hindrance of an amino acid loop. This has been a true deep dive. Thank you for guiding us through it.
It's always a pleasure to unpack these incredible mysteries.
And to you listening, thanks for trusting us with your time. We'll see you on the next deep dive.